All right, so this was originally intended to be the second half of a lecture on circulation and thermoregulation, the circulation part. But as it turns out, I had memory issues, not my memory, computer memory issues, and I had to split up the thermoregulation into two halves. And so this is going to be a new lecture on circulation, maybe respiration. It depends on how much stuff I've got going on after 30 or 40 minutes. And so we could tie the existence of circulation and circulatory systems as the result of a need to distribute material throughout the body of an organism. And so you could say that because animals are multicellular and generally pretty large, the need for a circulatory system goes part and parcel with our existence as animals as fairly large multicellular organisms. And one example that we've talked about as a mechanism for distributing food throughout the body of a sponge are the amoebocytes. And so you could think of that population of cells as serving as basically a form of a circulatory system. You might remember I called them the Uber Eats in the body plan of a sponge. Now, in the case of cnidarians like sea anemones and flatworms, the gastrovascular cavity, the digestive space, basically serves dual purpose as a digestive cavity for the breakdown and absorption of nutrients, as well as the distribution of said nutrients to the entire body of the animal. Like in the body plan of a sea anemone, food is going to be trapped in this area here, where digestion is taking place and absorption of the nutrients is going directly into the body plan of the animal. And so there's really no need. There's no need for a circulation in the rest of the animal because we can pretty much rely on simple diffusion to transport that material throughout the entire body of the animal. Okay. In the case of a flatworm, I'll draw a flatworm for you here. Flatworms also have gastrovascular cavities. Here are the eye spots. There's only a single tube. There's a pharynx, a little tube that comes out at the bottom of the flatworm. It attaches like right here in the middle, and that serves both as the mouth and the anus. The space that connects with the pharynx, that connects the outside world through the pharynx, goes into a pretty complex labyrinthine gastrovascular cavity. If I draw this from the top, it might look like this. Okay, and so here's where the pharynx would be. And from the pharynx, you would have a gastrovascular cavity that extends upwards. It's actually considerably more complex than that. But the idea here is that we have pretty much all the tissues in relatively close proximity to at least some space within the gastrovascular cavity. So as long as we have a uniform distribution of food within the gastrovascular cavity, there aren't any parts of the flatworm that will be so far removed from the food source that they would be nutrient deprived. Now, in the case of nematodes, we've got a pseudocoelomic space. Remember, that's an actual void in the body of the animal outside of the gastrointestinal space. In the case of nematodes, you've got a complete GI tract. So yeah, you've got a mouth and an anus, and the food goes from one end to the other. There's a little muscular pump at one end, but apart from that, most of the GI tract is just endodermally derived stuff. And then outside of that, you've got a muscular body wall. And the space in between the body wall and the GI tract is the pseudocoelom. And that's ultimately the space in which we'll find a circulatory system, a true circulatory system evolving. But in the case of a nematode, we don't have a true circulatory system. All we have is kind of like a redistribution of material that is absorbed into the pseudocoelom. It's basically put into the pseudocoelom, and the fluid that's there in the pseudocoelom cavity is distributing the material to the entirety of the nematode's body. Okay. Think about that as a starting point for the evolution of a true circulatory system. Because if we were to start with something like a nematode's body plan, and remember this here is a pharynx, a muscular pump, that pushes the animal's food down the digestive tract. It creates positive pressure and results in bulk flow down the remainder of the GI tract. If we were to install some type of secondary pump inside the body cavity, and maybe we'll have another pump, kind of like the pharynx over here, and that pump is going to be circulating, it's going to be pushing coelomic fluid in one direction only. That would have the effect of increasing the rate in which the coelomic fluid would be able to deliver nutrients and respiratory gases to the entire body of the animal. This extra pump that is circulating the coelomic fluid is basically serving as a sort of a heart. And effectively, that's not all that far removed from the most fundamental of our true circulatory systems, one of which is going to be the open circulatory system, and the other one is going to be closed. We'll talk first about the open circulatory system. And I'll make the case that an open circulatory system is basically exactly what we have here, only with a little bit more elaboration. So the animal I want to draw for you is a crayfish. Okay, so crayfish looks like this. So there are the antennae, the eyes, and the GI tract of a crayfish kind of looks like this. You've got a mouth over here, the esophagus goes all the way up to the top, and there's a stomach at the top of the animal, right? And then the intestinal system is going to carry the food or what remains of the food all the way back to the end. Okay? And that's where the waste comes out. And so the food goes in here, comes out on the other side. Okay? We also have claws, we've got callipeds, we've got walking legs, and that's basically the main body of the crayfish. Okay? Now, the circulatory system of the crayfish consists of several hearts, actually 3 or 4 hearts, and these hearts are connected through an aorta, right? And so these hearts pump the blood forward, and so the blood circulates forward, and eventually all the blood is released in the anterior or the head region of the crayfish. It's basically released into the general body cavity. We call it a general body sinus, and it openly flows. It flows backwards, 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 backwards through the entire body cavity of the animal, and ultimately it flows back in through one of these pores on the hearts, and then it circulates back to the front. The legs will push the blood through a series of gills. So you also have a set of gills, and so the hemolymph passes into these gills where it picks up oxygen. Okay? Water is circulating through the gill cavity, and so we basically have 2 kinds of hearts. The actual hearts that are along the main axis of the animal, that's pumping the blood from the back to the front, and you also have the legs, the leg muscles, which are functionally hearts because they push the blood through the gills. Okay? But in effect, what we have for the open circulatory system of the crayfish is nothing more than what we have up here. Okay? Hemolymph flows generally through the open body sinus, gets picked up in a vascular system. It gets picked up in some type of circulatory system, at which point it turns into blood. As long as the stuff is inside of a vessel, we call it blood. When it's in the open body sinus, we would call it hemolymph. So in the case of a crayfish or any animal with an open circulatory system, the blood really is the same as a hemolymph. It's just a matter of whether or not it's in some type of vessel. Right? And that's the same as the coelomic fluid. There's no distinction in the circulating fluid as far as the basic composition. Now, in contrast to an open circulatory system, the closed circulation is somewhat more complex. It basically requires the separation of the circulating media into 2 separate compartments. You've got the blood, which stays inside of the vessels, and then you've got the coelomic fluid, which is outside of the cardiovascular system. So the open circulatory system, in a certain way, it is deriving from simply just a secondary pump that circulates the coelomic fluid, kind of like what we have in the case of a nematode. You can say that the open circulatory system is the starting point for the evolution of the closed circulatory system, in which there's a complete separation between the blood and the fluid and the rest of the body cavity. I'll try to draw that for you in a simplified way. So if you want to simply contrast this with the open circulatory system that we have over here, you would basically say, well, it's the same thing. We've got a heart, and it pumps the blood forward, but instead of flowing out into an open body cavity, it's going to remain in a blood vessel. And that blood vessel is going to communicate with other blood vessels, and you'll have these larger blood vessels, the arteries and the veins, and you've got capillary systems that transfer the blood from one to the other. You could basically have a circulatory system where the blood is moving forward from the heart through these capillary systems into a venous system, and the blood eventually returns back to the heart. And because the blood is never leaving the vessels of the circulatory system, that's what makes it a closed circulation. So to put this into perspective of a more complex real animal, think about a fish. I think we might have even seen this already in the laboratory. You've got a heart down here. This is the ventricle, and the heart pumps the blood forward. Remember the bulbus arteriosus, and then we've got the gill arches. and the gill arches convene at the very top. And so you've got the arterial system that carries blood away from the heart, and then you have a system of veins that carries blood back to the heart and eventually comes back into the atrium. Okay, so the atrium is here. We also saw in the case of our fish dissection a large area over here, the sinus venosus. And so out in the body of the fish, we've got the capillaries, what carry the blood from the arteries to the veins. And so in the circulation of the fish, the blood never leaves a blood vessel. And so a fish is a good example of an animal that has a closed circulatory system. And so there's this general thing that biology teachers do sometimes, which is to try to evaluate or compare open versus closed circulatory systems, as in, like, well, which one is better? It's not really a valid thing to do, but we're going to do it anyways. And so we can talk about advantages versus disadvantages for open versus closed circulatory systems. Now, in the case of open circulatory systems, the general advantage is that this is a simple and direct system. We have direct contact between the circulating media, hemolymph or blood, and the tissues and the cells that are going to be served by the circulating media. The disadvantage of open circulation is that it's relatively slow. As the hemolymph is flowing very sluggishly back through this open body sinus, we can't really count on very rapid delivery of material to the tissues. It's definitely a whole lot slower compared to the circulation in something like a fish, where you've got a closed circulation. And so over there, we might as well say, well, in the case of a closed circulatory system, the primary advantage is that we've got a very fast delivery of nutrients and oxygen to the tissues. Speed counts. The disadvantages include the indirectness of the delivery of materials, the goods, to the actual cells. For example, if I got my blood vessel over here, the blood is actually moving through that blood vessel relatively quickly. That's the good part, right? So you might have red blood cells that are flowing through the capillaries and they're just tumbling along. The cells that are being served with oxygen and nutrients are out here. And so a nutrient that is circulating in the bloodstream has got to first diffuse into the space outside of the blood vessel, and from there it's going to have to diffuse into the actual cells, right? And so you've got a two-step process. Both oxygen and nutrients have to pass through two steps, from the blood into the interstitial spaces, and then from the interstitial spaces into the cytoplasm of the cells, in contrast to what happens in the case of open circulation. So these are the cells to which the circulatory system will be delivering nutrients. The nutrients are flowing through the hemolymph, which is directly in contact. So if the flow of hemolymph is in this direction, we don't have to go through two different steps. We can basically serve those cells directly with the nutrients or with the gases. Okay? So in contrast to this direct delivery system, the indirect delivery of closed circulation can be viewed as one disadvantage. The other disadvantage is that this is complex compared to the simplicity of an open circulation. There are lots more pieces that are required. There's a lot greater investment that the animal needs to make in order to have closed circulation. And we'll see some evidence of that a little bit later. Another way of dealing with this comparison is to use the comparative approach, comparing organisms that have open circulations versus animals that have closed circulations. And so if we look at the kinds of animals that have open circulations, we're talking about most types of invertebrates. All arthropods, gastropods, and bivalves, both of those are mollusks, remember? And on the closed circulatory side, we've got all vertebrates, we've got the cephalopods, and most annelids. And so one thing that some people might be inclined to do with using this comparative approach is to compare arthropods with vertebrates. After all the arthropods, crustaceans, insects, spiders, the chelicerates, they've got open circulatory systems, whereas all vertebrates have closed circulatory systems. But the problem with that doesn't work very well, because it really is like comparing apples with oranges. If all apples have open circulatory systems and all oranges have closed circulatory systems, you're not really able to evaluate open versus closed, because we're really talking about two different contexts. Okay. On the other hand, if we look within the mollusks, we have cephalopods with closed circulatory systems and gastropods and bivalves with open circulatory systems. And so in this case, it's kind of like comparing apples with open circulatory systems, gastropods, and bivalves with apples that have closed circulatory systems, right? The cephalopods. Similarly, we can make a comparison between the majority of annelids, which have closed circulatory systems, with leeches, which are annelids that have open-ish circulatory systems. And so we're going to be doing this comparative analysis. First, looking at those mollusks that have open versus closed circulatory systems. Now, thinking back to a laboratory, maybe last week or the week before, we thought about molluscan evolution. And from a very early common ancestor, maybe in the latest part of the Proterozoic, maybe in the earliest Cambrian, there was a most basal branching, one lineage from which gave rise to bivalves, and the other one relatively quickly split into the cephalopods and the gastropods. Modern bivalves and gastropods having open circulation, and cephalopods having closed. And we could ask a basic question about this molluscan Merca, most recent common ancestor. Did it have open circulation or did it have closed circulation? And thinking about this, there are two separate narratives that have to be true if you have one scenario or the other scenario. For example, if that common ancestor had closed circulation, it would have to have evolved closed circulation from presumably the ancestral state, which was open, some kind of open ancestor, gave rise to a closed molluscan Merca. And from that closed molluscan Merca, we would have to have two more state changes, one occurring right here, going from closed back to open, and one occurring over here, going from closed back to open in the case of gastropods. Okay. So in other words, the story, the narrative that's required if the molluscan most recent common ancestor had closed circulation requires three character state changes, going from open to closed or from closed back to open. All right? Now let's consider the alternative. If the ancestor had open circulation, then there's really only one character state change required. I mean, we could presume that the molluscan ancestor retained the open circulation of its more primitive ancestor. Remember that between open and closed, open is more likely the ancestral state and closed is more likely derived. So basically, if we had the ancestral or primitive state of open circulation at the most recent common ancestor, then there's only one character state change that's required. It would occur right here along the cephalopod lineage, starting off with an open ancestor and giving rise to a closed circulation character state for the ancestor of all modern cephalopods. Okay, one character state change required, and this is far more parsimonious. Now remember, Occam's razor says that the more simple explanation for a history, in this case, one character state change is simpler than three character state changes. A more simple explanation is more likely to be the true one. Okay, but there's definitely more to this story. I mean, if we think about cephalopods, they're predators. They hunt down, they chase down prey items, whereas bivalves are filter feeders. Gastropods, they're mostly herbivorous grazers. They crawl around and scrape algae from the rocks as they move along the bottom. Of these three ecologies, predation is the one that has the greatest demands for high metabolic rates. Remember, speed, intelligence, strength, these are things that are probably more important for predators than they are for their prey items. And closed circulation is providing oxygen to the tissues at a much faster clip compared to open circulation. So the basic idea behind this particular narrative is that within the molluscan clade, okay, you pretty much have everybody with open circulation. It's a characteristic that kind of is there for all of mollusks, with the exception of this one group that took on a predatory ecology. They're under extreme pressure to elevate their metabolic rates, and the evolution of closed circulation occurred as a result. Now, the annelid story is going to be a little bit more complex. From the annelid most recent common ancestor, we've got a basal lineage called the Arantia, and they have closed circulation. Right? These are the clamworms and bloodworms that kind of look like centipedes. They don't have jointed appendages, but they do have segmentation, and they crawl through the sediment. They crawl along the bottom. They get into bivalves. They do a lot of things. They're actually pretty common in fouling communities. Right? The other lineage is called the Sedentaria. Errant, like they're running errands, sedentaria, like they're mostly sedentary. And one group of the sedentaria are the tube worms. These are the worms that live in tubes. They have closed circulation as well. And a tube worm typically has a little set of appendages, parapodia, and bristles that it sticks out, but it lives primarily within a tube. Okay? And these are marine worms. We find them pretty much all over the world. The other lineage of Sedentaria includes the Clitellata, which includes both earthworms, and they have closed circulation. Remember, most of the annelids have closed circulation. And then we've got the leeches. And leeches are kind of special among annelids because they tend to have, as I said, an open-ish circulation. Okay. And the question here is, how do we explain this arrangement? Okay. You could ask the question, the annelid common ancestor, did it have open or did it have closed? Which was it? It can't be both. Okay. And I'm going to make the argument here that the common ancestors of the annelids actually did have closed circulation. Basically, from whatever ancestor that had the primitive state of open, there was a transition that went on. It gave rise to a closed circulation in the annelid most recent common ancestor. And that closed circulatory system has basically remained the characteristic for pretty much the entirety of the annelid clade. And there's one kind of renegade group, the leeches, where we've had a transition from the closed circulatory system that characterizes most of the annelids over to an open circulation, which is what we find in many leeches. Okay. And the argument goes like this. It's actually kind of a neat argument because it makes us think about something we've already talked about. And so you might remember from an earlier lecture, we talked about the coelomic space as having a function as a hydrostatic skeleton. Remember that? The muscular walls of the worm have to push on something in order to make the worm move. And so having this enclosed space in the body of the worm, which is otherwise a pretty muscular wall, needs to be able to compress the space, causing the worm to either extend, make it longer. Basically, if we contract these circular muscles like this, we've got circular muscles. When we contract those muscles, the worm gets longer and skinnier, and then we've got longitudinal muscles that go in this direction all the way around the worm. And when these muscles contract, the worm gets shorter and fatter. Okay. That was a basic argument of a hydrostatic skeleton. Okay. Now, annelid hydrostatic skeletons are operating on pretty much the same principle, with one exception. Instead of having a contiguous hydrostatic skeleton, the coelomic space is segmented into completely isolated compartments. We've got these walls, they're called septa, singular septum. And all these septa isolate the body cavity into separate compartments. So there's the muscular wall to the worm, and the advantage of doing things this way is that each coelomic compartment can be acted upon independently by the musculature. You know, for example, if we just take this little coelomic space right there, if we were to contract the circular muscles around that space alone, we would be able to cause this compartment to stretch out and become as long and thin. We can alter the shape of that fixed volume of non-compressible fluid in that first coelomic compartment independently from everybody else. Okay? On the top worm, if we were to contract the muscles in the front portion of the worm, that would have the effect of pushing a lot of the coelomic cavity towards the rear. In other words, we wouldn't really be changing the shape so much here. We wouldn't be able to extend that out as effectively as we do in the segmented worm. And that's kind of an oversimplified picture of why a completely septated coelomic cavity makes for a much better hydrostatic skeleton. But I hope you can get the picture, right? And so the flip side of this, the consequence of having a completely septated coelomic cavity in order to make this super effective hydrostatic skeleton is that it makes it practically impossible for us to use open circulation. We can't pump all the blood into the front compartment and expect it to flow backwards. There are these septa in the way. And so the only way for us to get complete circulation is to have blood vessels that carry the blood all the way through the worm. And so we have pretty much a closed circulatory system in the case of a worm. We'll have hearts around midline. And we'll have that situation where the blood flows through these capillaries from one blood vessel to the other one. And my point here is that closed circulation is evolving in annelids potentially for a completely different reason compared to the mollusks. I mean, it's not that the worms demanded greater delivery of oxygen to their tissues. Now, to be honest, they got that anyways when they evolved closed circulation. But the main driving force for the evolution of closed circulation really had more to do with this hydrostatic skeleton and the advantages associated with having it completely septated. Okay. Now, the ecologies that favor this type of hydrostatic skeleton and its associated closed circulation are those of burrowing or crawling worms. Okay? Now, leeches, in contrast, it turns out that they change their ecology away from burrowing or crawling. They don't need to have this super effective hydrostatic skeleton. They tend to swim or creep along the bottom of stagnant water, or in the case of terrestrial leeches, they tumble. Okay? If you ever go to India and walk through the forests, you'll have the tremendous joy of encountering leeches that will catch into your socks and start sucking on your blood as you're going for a walk in the woods. Now, leeches have lost their internal septation. They look segmented from the outside, but inside we don't have those complete septa that go all the way across, right? And so the body cavity of the leech is more or less contiguous. And that's actually a good thing for a leech for another reason. Okay? Remember that leeches are fluid feeders. They'll take in a really large blood meal. So an unfed leech will have its GI tract relatively narrow. But after this leech has a chance to feed on a host for quite a while, it's going to fill the space with a large volume of blood. Okay? And taking that into account, you can see another reason why leeches would actually be inhibited by having complete septa. If we had these septa that went all the way across the body of the leech, it wouldn't be able to take in that enormous blood meal. It'd be kind of like trying to eat a huge meal while you're wearing really, really tight clothes. These internal septa would prevent the leech from taking in as great of a volume of a blood meal as it could in the absence of those septa. All right. So for those reasons, you could sort of say, well, leeches gave up on hydrostatic skeletons, which they probably had in the past, and not having that constraint on the circulatory system. They don't need to have a closed circulation anymore. And they've partially reverted back to an open circulatory system. The upper quadrants of their coelomic tract are now kind of like the functional hearts for the leech. The muscular contractions of the leech basically push the blood through what amounts to an open body sinus. So in the case of mollusks, we have cephalopods that seem to evolve closed circulation primarily in response to selection for higher metabolic rates. The greater need for intelligence and locomotory performance in these predatory animals demanded that they have a greater delivery of oxygen to their tissues. Now in the case of annelids, closed circulation evolves in response to the worm's need to take full advantage of this hydrostatic skeleton. Okay? Now, there's one group of animals that seems to defy this basic logic, and that's the insects. Insects have both open circulation as well as extremely high metabolic performance. They're capable of powered flight, which is one of the most intensive things that any animal can do. So, on the face of it, this seems to be a paradox. It seems to fly right in the face of our earlier arguments. Okay? But it's not that difficult of a thing to explain. Okay, if you look at the structure of an insect, I'm going to draw kind of a detailed fly for you. And the flight muscles are in this area right here. And we've got other musculature elsewhere. We've got the muscles in the legs and the rest of the body. The thing to look for the next time you come really close to an insect are little tiny holes that are there along the outside of the exoskeleton. These tiny holes are called spiracules. Okay, which literally means little spiracles. A spiracle would be a tiny hole through which the animal is respiring. Cule basically makes it smaller. Now, the spiracle connects the tracheal system to the outside air. And each tracheole connecting to a spiracle starts to branch out into finer and finer small tubules that contain air. And the very tips of the tubules, you have the muscles that are requiring oxygen and other things. Basically, you've got the tubules that are basically serving these muscles with air directly. And so the mechanism of delivery of oxygen to these tissues doesn't really require any circulation at all. All the muscles, which are the most oxygen demanding tissues in the insect's body, are relatively close to a supply of oxygen. That's the outside air. We've got 21% oxygen in the ambient air, and that oxygen has an opportunity to diffuse through these tiny, tiny tubules to get all the way out to all the tissues of the insect, right? So basically what I'm telling you is that insects don't need to rely on their open circulation for the delivery of oxygen to their muscles because they have this tracheal system, right? They're basically feeding their flight muscles with oxygen via diffusion over a relatively short distance. You could say that one of the constraints that prevents insects from ever becoming too large is that it could put the flight muscles at too great of a distance in order for this diffusive mechanism to work. Okay, so it could have been that those huge dragonflies that we had during the Carboniferous were taking advantage of the elevated oxygen levels during that time. Remember why we had elevated oxygen levels during the Carboniferous? Well, we also had huge dragonflies. And what I'm saying here is that, if we have a modern constraint, if there's a modern upper limit to the body size that insects can be and still use diffusion to serve their muscles, that upper limit would have been different in a world in which we had 28 or 30% oxygen.